All right, so now given that it's post-lunch and I'm the only one standing between you and the coffee break, the last thing you need to hear about is sleep. But I hope you'll bear with me for the next half hour or so. So sleep is a process that's seen in organisms of, this is the laser, okay, not that one. Organisms of many different species, as you can see here. Sleep is controlled by two major systems, the circadian clock and then the homeostatic system. Now, the circadian clock controls the timing of sleep, ensures that it occurs with a 24-hour rhythm. But even if you had no circadian clock, you would still sleep. It just wouldn't be with the 24-hour cycle. So the occurrence of a sleep state, the amount of sleep, in other words, is really determined by this homeostatic system, which drives the need to sleep and ensures that an organism gets enough sleep. And that is because sleep serves one or more essential functions. We still don't really know what those functions are, even though we spend a third of our life sleeping, or we should spend a third of our life sleeping, I should say. But we do know that it is very important. Now, typically, these two systems act together to determine when sleep occurs, but sometimes one can override the other. So if you've been up all night, you will sleep in the morning, even though your circadian clock at that point is telling you to wake up. And that's the homeostatic system at work. Now, we use the fruit fly, Drosophila melanogaster, to understand the control of sleep by both these processes. And what I'm going to do today is sort of quickly summarize, in a general sense, what we know about the circadian regulation of sleep, and then spend more time telling you about our efforts to use the fly to understand homeostatic regulation. So work done in several labs, including ours, has led to a basic understanding of how circadian clocks are generated. In flies, in its very simplest form, the circadian clock consists of this molecular feedback loop, shown here in a single cell, a clock cell. At the heart of this loop lie the period and timeless genes, which are transcribed rhythmically. They were transcribed during the day. The two proteins build up at night, go into the nucleus, and inhibit the activity of transcriptional activators, clock and psych. When PER and TIM are degraded in the mornings, this loop resumes. Now, for the longest time, when we showed this loop, we said this is thought to be the underlying basis of behavioral rhythms. At this point, I think it may be fair to say that this is the underlying basis. I don't know, maybe there are some people who still don't agree. But what's really important here is cyclic expression and activity of these two proteins. And we've actually put a lot of effort into trying to figure out what it is that needs to cycle, and this is really what needs to cycle in the Drosophila clock to drive behavioral rhythms. Now, so the idea in its very simplified form is that the levels of these proteins are telling the animal what time of day it is. So if that's the case, then you would expect that anything that sets the time of this clock would do it by changing levels of these proteins, and that indeed is what light does. So we showed several years ago that the timeless protein gets degraded by light. In more recent years, we found that light signals are transmitted to timeless by the circadian photoreceptor cryptochrome, or CRY, discovered by the Hall, Rosbash, and K labs. And we discovered this molecule jet lag, or JET, which is an E3 ligase that is required for this process. Now, in order to maintain this loop and get a 24-hour period out of it, you have to have delays built into the cycle. Many of these come from post-translational modifications, phosphorylation being the most important one. So shown here are some of the kinases and phosphatases that regulate PER and TIM. The proteins are phosphorylated rhythmically, and it appears that the rhythmic phosphorylation really comes from rhythmic activity of phosphatases rather than kinases. Now, it turns out that these mechanisms and most of these molecules are very well conserved. So here is the central clock mechanism in mammals. I won't go into details here. I think you'll note that most of the components are the same. The mechanisms are the same, to the point where the kinases and phosphatases are also pretty much the same. And we now know also that components of this loop are mutated in humans with circadian disorders. So work done in the Potachik and Fu labs has shown, they've been studying people with advanced sleep phase syndrome. This is a circadian disorder in which people have cycles that are advanced by several hours with respect to the rest of the population. So they go to bed at 7 p.m., wake up at 3 a.m., and in one such family of people with this disorder, there's a mutation in the PER2 gene, and in another one, there's a mutation in CK1. Both genes that were first found in flies as components of the circadian clock. So there are many aspects of the clock that we still don't understand. I don't think anybody would try to claim that it's solved. There are probably 50 to 100 other genes that are going to end up being part of it. But thus far, it does seem that the basic mechanisms are conserved. And we've put some effort also in figuring out the pathways that transmit time-of-day signals from the clock and drive rest activity rhythms. And some of those are shown here. And some of these also end up being conserved. So Fragile X, for instance, or FMR, we and Tom Youngins showed is required for circadian rhythms in flies. Turns out to also be the case in mammals. The RAS-MAP kinase pathway actually working together with the neurofibromatosis 1 gene. And the RAS-MAP kinase is also relevant for the mammalian clock. All right, so the reason I'm sort of stressing the conservation here is because this is one of the things that spurred us into trying to use the fly to address the homeostatic regulation of sleep. So this was work we did about 10, 12 years ago. Up until that point, we had really just been studying circadian rhythms in flies and using the rest activity behavior as a readout of internal clock function. So we weren't necessarily interested in the behavior per se. The behavior was just a way of measuring the clock. But then we became interested in whether rest in flies is a sleep-like state. And as I just mentioned, one of the reasons was because we had noticed that on the circadian end of things, mechanisms were conserved. So we thought, maybe they would be for the homeostatic control. The other thing is that at the time, there was this growing awareness that we know very little about sleep, and we need to do more basic research, and perhaps model organisms could help this effort along. So I'll summarize what we did, or rather what Joan Hendricks, who was a sleep researcher who came to my lab to do a sabbatical, did. And what she did was to determine if fly rest is a sleep-like state, basically to determine if it meets criteria that have been proposed for sleep over the years. Now, the fly does not really lend itself to EEGs, so we steered clear of those and try to satisfy other criteria, and those are shown here. So we already knew it was a reversible state of behavioral quiescence. We knew that it occurs with the 24-hour rhythm. When flies are sleeping, they show resistance to sensory stimulation, so they have an increased arousal threshold, a hallmark of sleep. And we knew that, and then Joan showed that fly sleep is regulated by homeostatic mechanisms. In other words, if you deprive them of sleep, they will make up for the sleep lost in the morning. They show a robust rebound. And that can actually be seen visually. This thing works. Oops. Okay, so what you're looking at here are two bottles of flies. The ones on the right are control flies. They slept at night. This is recorded in the morning. They're very active. The ones in the bottle on the left were sleep deprived at night. And so they are undergoing their sleep rebound. I know they look dead, but trust me, they're not. Okay, so now here are some additional features of Drosophila rest that are reminiscent of human or mammalian sleep. There are age-specific and gender-specific patterns. I said that we didn't want to do EEGs, but in fact, Ralph Greenspan's lab did the, as close as you can get, equivalent of EEGs in the fly. They recorded from fly brains and showed that electrophysiologically, the patterns are different during sleep. So again, I don't think that they would use this routinely as an assay, as we wouldn't, but they've nevertheless established that there are electrophysiological changes, and there are molecular changes. So somebody earlier here today mentioned inflammatory markers changing as a result of sleep state. That's actually true in the fly as well. So when we've done gene expression studies, then with sleep deprivation, you get an increase in inflammatory immune markers, just as you do in mammals, and in humans too, actually. So, and then fly sleep is responsive to drugs that affect human sleep. And I'm showing you here data for a drug called Modafanil, commonly known as provigil, which is quite a popular drug these days because it helps you stay awake, but unlike caffeine, it's not a stimulant where when the effect wears off, you have a huge rebound. And so for that reason, I think people tend to take this a lot. So modafinil maintains wakefulness in flies. These are activity records from 3 flies maintained on modafinil and 3 controls at the bottom. And so each day here, each line is 2 days. And you can see that these flies that were maintained on modafinil the first few days are almost constantly active. At the arrow shown here, modafinil was removed, and then they go back to their normal sleep-wake rest activity pattern. And the same thing is true. I'm sorry, these are 3 controls that maintain this similar normal rest activity pattern all throughout. And as I mentioned, these cycles deteriorate with age. So here is a young female fly, very robust pattern of rest activity or sleep-wake. And here's the same fly when she's old. And you can see that the rhythm falls apart. So you start to get more activity at night, and you start to get more daytime sleep. The same is true in humans, of course, although with humans, unlike the flies, you can't monitor the same subject all throughout. So here are data from a young subject over a 5-day period, very robust activity patterns. These are people that were subjected to actigraphy, which Dave talked about earlier today. Here is the older subject that has increased nighttime awakenings and in more daytime sleep. Now, I'm not going to spend a lot of time on this, but actually we've recently put a lot of effort into trying to figure out what happens with age. And what I'll tell you is what doesn't happen, and that is that the central clock does not fall apart. So the central clock that drives rest activity rhythms is actually very robust in old animals, even in the ones that look completely arrhythmic. So we've actually tested them for behavior first, taken those flies, and then stained for clock proteins, and the cycling in the central brain is very robust. In peripheral clocks, so clocks, I mean, I'm focusing today on sleep, wake, of course, but many of you may know that there are clocks in pretty much all body organs, the liver, the kidneys, the pancreas, and so forth. And those clocks tend to dampen with age, but the central clock in the brain does not. So whatever happens to the sleep-wake cycles with age occurs downstream of the clock. And we have reason to believe that the same thing is actually true in mammals. So what we want to do with this, I should mention, we have established this fly model for sleep, and at the same time it was established by Paul Shaw in Giulio Tononi's lab, and there are now dozens of labs that work on fly sleep around the world. And I think what we all want to do is get at the cellular and molecular circuitry that underlies sleep. And with respect to that circuitry, the first thing we found when we tried to identify regions of the fly brain that were important for sleep is that this particular structure shown here in pink, the mushroom body, was important. So this is the fly brain here. Here's the optic lobes. These are the circadian clock neurons shown circled here. And then this is the structure, the mushroom bodies, that we found was important for the homeostatic control of sleep. So if you manipulate mushroom bodies, you get changes in the amount of sleep. You can get decreases or increases based on which part of the mushroom body you target. You don't get changes in the circadian pattern. So on a cellular level, you can actually separate these two types of control. Now, one of the things that was interesting to us about this is that the mushroom body is also a seat of learning and memory in the fly brain. So these findings lend support to the idea that, and I know Matt is very familiar with this, which is that one of the functions of sleep is to consolidate memory. So in our efforts to identify molecules, I mentioned that we've done differential screens to look for genes regulated as a function of behavioral state. We've also tested candidate genes, and some of these are shown here. So the usual suspects: dopamine, GABA, norepinephrine, which in flies is octopamine, serotonin, cyclic AMP signaling, and EGF signaling all have effects on wake and sleep in flies and in mammals. Many of these were known to affect sleep in mammals before we looked at them in flies. The cyclic AMP work we did at the same time as Ted Abel, who showed that in mice it promotes wakefulness. And in recent years, actually even over the past year in particular, there's been a lot of progress in figuring out what cells are mediating effects of these different neurotransmitters. So here's a schematic of the sleep-regulating cells in the fly brain. Now, the structure here in red is the one I pointed to earlier, which is the mushroom bodies. This one here, right at the dorsal tip, the green and blue, is, I guess you could call it the fly hypothalamus. It's a bunch of neuroendocrine cells, that some of which promote sleep and some of which promote wake. And then here are the clock neurons. Now, we found that serotonin promotes sleep consolidation, and it does so by acting through the mushroom bodies. Octopamine, which, as I said, is fly norepinephrine, promotes wakefulness. Now, there are many octopaminergic neurons in the fly brain. Through a mosaic analysis, we were able to show that it's these in the dorsal part of the fly brain that are really important for the effects on sleep-wake. And if you activate just one of these octopaminergic neurons, you actually get significant wake-promoting effects. And the target of these octopamine neurons is a specific octopamine receptor, the OAMB receptor, that acts in this fly hypothalamus, or pars intercerebralis, as it's called. Mark Wu's lab showed recently that the source of the wake-promoting dopamine is the PPL3 neurons. Again, there are many dopaminergic neurons in the fly brain, but these are the ones shown here that are important for promoting wake. And again, the activation of a single neuron has a significant effect. And the effects of dopamine go through this structure here called the fan-shaped body. This was also shown by Mark's lab. Now, the circadian clock neurons are kind of interesting. The small ventrolateral neurons shown here, the little blue ones, they are these central clock neurons, and they're absolutely essential to maintain a rhythm in the absence of environmental cues. So they really drive the rhythm. The large ones shown here really, as far as we could tell, didn't have a role in circadian rhythms, even though they express clock genes. And we now know through work done in the Ross-Bash and Griffith labs that they are required for light-induced arousal. They're inhibited by GABA, and they also receive dopaminergic signals. So we're starting to sort of get the cellular circuitry together here. Now, dopamine has actually really interesting effects because in addition to its wake-promoting effect that goes through the fan-shaped body, it reverses the phase of activity. So flies are normally diurnal. And so shown here is a histogram. depicting activity and rest in a population of flies. And you see that most of it is during the day. There is a dip in the afternoon, an afternoon siesta. They anticipate lights off, lights on, but most of the activity is during the day. In flies that have high dopamine signaling, like in this one mutant shown here, you actually get nocturnal behavior. And so this was something that we reported very recently, and the way to really see how this happens is to look at flies not in a 12-12 light-dark cycle, but in a 4-4 light-dark cycle, where you get rid of all circadian influences and you're looking at just the light or dark-driven event. And in this type of cycle, the wild-type flies show startle responses to lights on and to lights off. In these flies with high dopamine, on the other hand, there's no startle to lights on. It's only to lights off. And we mapped this effect of dopamine and found that it requires the large ventrolateral neurons. It actually, which are these large, the cells that normally produce light-dependent arousal, in the presence of high dopamine, promote dark-dependent arousal. And that particular response requires the circadian photoreceptor cryptochrome, or CRY. But we believe that in this context, cryptochrome is not functioning like a photoreceptor. It's actually known to modulate neural activity, and that was shown by Todd Holmes's lab last year. And so typically what cryptochrome does is, in response to light, it promotes neural activity in these cells. But when you have high dopamine, cryptochrome is required to promote the activity at night. And I won't go into why we think that is happening. If anybody's interested, we can discuss it later. So, so this is the circuit mapping as it stands for the known neuromodulators that affect sleep in the fly. Now, the reason, of course, that we chose to develop the fly as a model was not to take known neurotransmitters and figure out how they act, but to discover new molecules. Because given that sleep was a process about which nothing was known, we felt that the unbiased approach, which the fly allows through genetic screens, would be the best one to get at new things. So we've been doing genetic screens for sleep mutants. And the first one we got was a mutation in the dopamine transporter, which was nice, kind of validated the screen, but didn't move us forward. And then shown here are data for another mutation that came out of the screen of about 4,000 lines. So sleep in these lines, the amount of sleep is normally distributed, and the arrow points to a specific, an extreme short sleeping mutant. And this mutant, for obvious reasons, we called sleepless. And you can see here for sleepless males as well as females that they have greatly reduced sleep. So here's a wild-type control male, a lot of sleep at night, a huge afternoon siesta. Sleepless males are reduced at both times. Females have much less of a siesta. That's typical in all strains. But they have very consolidated sleep at night, and again, both are reduced in the sleepless. And if you quantify it, sleep is reduced by 80% or more. Now, we cloned sleepless, found that it encodes a small protein that's present at very low levels in the body, if at all. It's in the head, enriched in brains. It is glycosylated. In fact, it's GPI anchored. And you can see that here in that when you treat it with glycosidase, the molecular weight goes down. Now, the original sleepless mutant slept so little that we couldn't do the homeostasis experiment. In other words, deprive it of sleep and look for rebound. But we were fortunate enough to get a second allele that has normal levels of daily sleep. So we did the homeostasis experiment with that. And so when you deprive this mutant of 6 hours or 12 hours of sleep, it loses the same amount as control because they have the same amount to begin with. But rebound is greatly reduced. So sleepless is required for daily sleep, and it's also required for rebound following sleep deprivation. So I should mention that this particular mutant is a hypomorph, so it has reduced levels of sleepless. So when you have reduced levels of sleepless, it's enough to give you normal daily sleep, but it's not enough to overcome the circadian drive in the morning and let you have rebound. If you actually put this mutation in a clock mutant background, you get a rebound. So when you get rid of the circadian drive, it's actually sufficient. So what does sleepless do? We looked to the literature for connections with other sleep mutants. The only other one known at the time was Shaker, which is a voltage-gated potassium channel shown to affect sleep in the Tononi-Cirelli lab. We found that sleepless maps very close to Quiver, which is a mutant that affects Shaker potassium current, but that had never been cloned. So in Quiver mutants, Shaker currents are greatly reduced. So we cloned sleepless from Quiver, found a single nucleotide change that alters splicing. So instead of the wild-type sleepless transcript, you get these 3 other transcripts, all of which we were able to detect. And one of these, Quiver2, is expected to encode a protein that's larger in size than wild-type sleepless. And indeed it does, but levels are greatly reduced. So sleepless and Quiver are allelic, and Quiver mutants also have a sleep phenotype. So that told us then that sleepless probably affects Shaker. And so we looked at the expression of Shaker in sleepless mutants, and we found that Shaker levels are reduced, as are sleepless levels in Shaker mutants, and that's here. So there is some reciprocal interaction here. Now, if you stain fly brains for Shaker, here it is in wild-type brains, very enriched in mushroom bodies. It's reduced in sleepless mutants. But more importantly, what you notice is that while in wild-type, Shaker is only in processes, so you don't really see distinct cell bodies, in the sleepless mutant, you start to see cell bodies here. So it looks like trafficking is also affected. So levels of Shaker are affected, trafficking of Shaker is affected. And finally, on an electrophysiological level, Shaker activity is affected. So we did these experiments, since we don't do electrophysiology in my lab. We did them in collaboration with Toshi Hoshi. And Terry Dean, a graduate student in the lab, found that sleepless and Shaker mutants have, so he used the neuromuscular junction to record from these flies. Not because this is a model for sleep, but because it just lends itself nicely to electrophysiology. And what he found at this NMJ was that MEP frequency was increased in sleepless and Shaker mutants. So neural excitability is increased. If you put sleepless back in the nerve, you rescue this increased MEP frequency, it goes down. If you put it in the muscle, you don't. So this is a presynaptic defect. So there's increased MEP frequency rescued by neuronal expression of sleepless. That's the presynaptic defect. We found, I'm summarizing a lot of work here, that there's delayed activation and reduced amplitude of Shaker current, and that's rescued by muscle expression of sleepless. And finally, we found through expression in heterologous systems as well as NMJ recordings that there are kinetic effects of sleepless on Shaker. So sleepless accelerates the activation of Shaker in the heterologous system. They both localize to lipid rafts, and the effect of sleepless on Shaker requires cholesterol. And finally, it reduces C-type inactivation of Shaker. So what was interesting was when we modeled the sleepless structure, what we found was that it looked very much like toxins, snake toxins. Shown here is a comparison with cardiotoxin. So the particular type of toxin that sleepless is, it's called a Ly6 domain containing toxin. And Ly6 domains are these three finger-like repeats that are formed through cysteine residues. And now for many years, people have speculated that there are internal toxin-like molecules that regulate channels, just like snake toxins do. And we believe that sleepless is one such toxin-like molecule. And I'm not getting into this today, but shaker is probably not its only target either. Now, the only other toxin I should mention, endogenous toxin-like molecule that's known, is a molecule called Lynx-1, which is also a Ly6 domain containing toxin like sleepless. And the target of that one is the nicotinic acetylcholine receptor. So our very simple model, then, is that sleepless is required to control neural activity and allow the sleep to occur, basically allow the homeostatic drive for sleep to be implemented. When you don't have sleepless, you have a big increase in neural activity, and this homeostatic drive cannot be implemented. So in other words, we believe that the sleepless flies are a genetic model of sleep deprivation. Now, as I mentioned, this is a very, very simplified model. In fact, we now know that this increased neural activity in sleepless flies is doing a lot of things, including affecting mitochondrial function and GABA transmission, but I won't belabor the details here today. So this basically are my conclusions, which I summed up in the model. And I just want to make one more point, which is that it does look like these mechanisms are conserved. So there is a group of syndromes. Most commonly, one is Morvan syndrome, where these people have neurological symptoms and, in addition, significant insomnia. And it turns out that they have autoantibodies to voltage-gated potassium channels. And a huge genome-wide association study done recently by a European group where they looked for SNPs that correlate with human sleep duration, found that a SNP in a KATP channel gene has an effect on sleep duration. They verified the effect of this particular gene by testing the homolog in flies. But what was really important to us was that one of the top hits in this study was a human ortholog of a regulatory subunit of shaker. All right, so I will stop here and just acknowledge the people who did the work. I tried to give credit as I went along. Most of what I talked about was sleepless, which was done by 3 very talented postdocs, all of whom now have their own labs: Kyung-hee Ko, Bill Joyner, and Mark Wu. Terry Dean did the electrophysiology with Toshi Hoshi. The work I talked about at dopamine was done very recently by Shailesh Kumar, a current postdoc. And I'll stop there. Thank you.
Are there vertebral mammalian homologs of sleepless?
So the bioinformatic, the comparisons have been difficult because it's such a small molecule. When you take away the GPI anchor and the signal sequence, it's less than 100 amino acids. But what we have done is identify all mammalian proteins in the databases that have the same type of structure, the Ly6 domain, small protein, GPI anchored. And we ended up with about 15 proteins that we figured we could test by an in vitro system for effects on shaker. And we got ready to do those experiments, and the student graduated. So hasn't been done yet.
So I'm not sure if you can test this very well on Drosophila. I'm just not familiar. But have you looked at any effects of learning and memory in the sleepless mutants?
So we haven't, because we don't really have expertise doing those assays. There are, but in fact, those, I mean, learning and memory can be tested in flies. And, we really just should ask somebody to do it, because I've been asked that question a number of times, and I wish I had an answer. I would guess that it's impaired. Their lifespan is cutting short. It's half that of, yeah.
Sorry, one quick question. Is
Sure.
the lifespan of the sleepless mutants affected?
Lifespan is reduced.
Just
Yeah,
reduced.
it's
By how much? Yeah.
about 50%
50%.
of normal. Yeah.
And do you see that in males and females?
We see that in males and females.
Females,
And that
right.
actually is seen for all sleep mutants in flies, or anywhere really, that short sleepers are short-lived, with the one exception being the flies that are mutant for the dopamine transporter,
Okay.
which
yeah.
actually have normal lifespan under normal conditions, but if you challenge them, then they're reduced.
Okay, good. Yeah.
Okay, one last question over here before the break.
Just a quick question. I agree with you. There's a striking homology between the foot fire and the mammals in their clock system, because I see, the both of them, the molecular circuitry, particularly the feedback system, are strikingly similar.
Right.
But I disagree with you. There's some divergence between the mammal and the fires. For instance, you mentioned about the cry.
Right.
System, I wonder maybe the cry 1 and cry 2 maybe lost its function as you go, across the, like just mouse or a simple rodent in the mammal system. And also I find that, in this two, system, you might transfer some of the foot fly study into the mammal, but you cannot do it system-wise. But you get the same
Sure.
genes cluster, but the translated, encoded protein, they may serve different functions. That's my impression.
So it varies from protein to protein, right? And so the specific case you brought up of cry, it's not just in mammals. There are actually many insects in which cry has, there are two cries, as you said, cry 1, cry 2, one of which is a photoreceptor, and one of which is just like the mammalian, clock transcription factor. So yes, there is divergence, not just from fly to mammal, but even within insects. And I think that you have to expect some amount of that to occur. But